This video will be control of gene expression in bacteria. This is the third video. And in this video, we'll have two topics. The first one is a second type of regulation that occurs with the lac operon. And the second is post-transcriptional regulation of gene expression. So we've talked quite a bit about the lac operon already, and we've talked about the things that were discovered by Jakob and Monod. The system of regulation that was discovered by Jakob and Monod involves the detection of lactose in the environment. And so this is a repression system shown over here on the right. And the idea of it is just that the cell is trying not to waste its cellular energy by making proteins that are not needed at a certain time. And so unless lactose is present, you don't want to transcribe and translate those genes. It's only when lactose is present that you actually need them. Now there's a second mechanism of regulation for the lac operon. This was actually not discovered by Jakob and Monod. The idea of this second regulation system is that it is a positive regulation system, and it involves the detection of glucose levels in the environment. So the overall idea of this is that a cell would prefer to use glucose if there were multiple sugars available. And that is because glucose can go directly into glycolysis. It does not need to be changed before it can be used for energy. This is in contrast to the sugar lactose. So the cell needs a way to sense the glucose levels in its environment. And in order to do that, there is a factor called cAMP. So cyclic AMP is actually, the structure is shown over here on the right, and it looks very much like a nucleotide if you notice. However, that phosphate is actually made into a little ring here. That's why it's called cyclic AMP. So we won't go into the details of why this is the case, but it turns out that cyclic AMP and glucose are oppositely regulated. And so when glucose is high, cAMP is low, and when glucose is low, cAMP is high. So it's opposite regulation. cAMP is going to be our cellular readout for the glucose levels in the environment. cAMP is a factor that will then bind to a protein, and that protein is called CAP. CAP, together with its cofactor cAMP, can bind to a region of the DNA on the lac operon right next to the promoter. When CAP binds to cAMP, it actually changes the conformation of that CAP protein, and the affinity of CAP for the DNA is greatly increased at that point. So it only binds to the DNA together with this cofactor, and then the function of that CAP cAMP complex is actually to help RNA polymerase bind to the promoter. So why would it be useful to help recruit RNA polymerase to the promoter? So RNA polymerase will bind to the promoter, any promoter that is not obstructed, for example, by a repressor protein like LAC-I. It will bind anyway, and so the use of CAP and cAMP is really to make RNA polymerase bind more rapidly to an empty promoter. This is actually helpful for the cell because once one RNA polymerase goes off and leaves the promoter and starts to transcribe, the time that it takes for a new RNA polymerase to bind to that promoter is really a limiting factor in how much RNA you can make from that particular operon. And so if you get RNA polymerase recruited very rapidly after that previous RNA polymerase leaves the promoter, then you're going to result in making very high levels of messenger RNA from that operon. In contrast, if you do not have CAP and cAMP to help out, it's going to take some time for RNA polymerase to bind to the promoter, and the result would be very little amounts of transcription, relatively speaking. So when you bring the positive regulation system together with the negative regulation system, you can actually have three different situations. One situation where you don't make any RNA and no transcription from that operon. The second situation where you just make a very little bit of RNA from that operon. And then the third situation where you make a lot of it. So in this first situation, if you have glucose present and you do not have lactose present, what would happen here? So glucose is high, therefore CAMP would be low, and CAP would not be binding to the promoter. Also, no lactose is present, and when you do not have lactose, the lac repressor will bind to the operator and physically block RNA polymerase from binding and transcribing, and the result would be that you have zero transcription. For the second scenario, if glucose was present, so if glucose was high, CAMP is the opposite. CAMP would be low. And in this case, CAP would not bind to the region near the promoter since it doesn't have its coactivator. If lactose was present in this case, lactose would bind to the LAC-I, the lac repressor, and make it fall off of the DNA. And so you don't have the activator, but you also don't have the repressor there. The result of that is that RNA polymerase would be recruited very inefficiently to the promoter, and you would transcribe occasionally. And so the result of that is that there's very low amounts of messenger RNA from that operon. The third situation would be that you do not have glucose present, so low glucose means that CAMP would be very high, and CAMP would then bind to that transcription factor protein CAP, and CAP then increases its affinity for the binding site near the promoter. CAP would then help recruit RNA polymerase very efficiently to that promoter. The second part here, lactose is present. Lactose makes the lac repressor fall off of the operator, and that frees up that region for RNA polymerase to bind. And so CAP is helping to recruit new RNA polymerase, and there's no repressor, and therefore you would very rapidly make more and more messenger RNA from that operon. The second part of this video will be looking at the post-transcriptional regulation of RNAs in prokaryotes. So the most common place that you would expect to regulate genes is in this decision of whether you transcribe that gene or not, whether you make the messenger RNA. There are many examples, however, where the messenger RNA itself is regulated, and this would be considered post-transcriptional regulation. We'll talk about three different ways in this particular video: antisense RNA, riboswitches, and ribozymes. So this first example, antisense RNA, I'm going to tell you about this gene called the OMP-F gene. So this encodes a protein that makes a channel for passive diffusion across the membrane. If the bacterium gets into an environment with very high osmolarity, it's going to actually suck all the water out of that bacterium. And you've really got to close off those passive diffusion channels very rapidly. So in order to do this, there's a couple of possible approaches. One way would just be to turn off that OMP-F gene so it's not transcribed anymore. But that is not going to give a really fast response. So there's still a lot of existing RNA and protein that's in the cell, and that cell might die, in fact, before it gets rid of all of it. So what the cell does is it turns on this functional RNA gene. It's called MIC-F, and this gene is actually complementary to the messenger RNA for the OMP-F gene, called an antisense RNA. And it binds to that messenger RNA and it prevents it from being translated. Riboswitches are another kind of regulation of RNA. So this is actually done by a regulatory protein. So this protein will bind to a messenger RNA and make that messenger RNA form into a particular secondary structure. When the structure is present, the ribosome binding site is actually hidden. So therefore that messenger RNA will not get bound by ribosomes and the gene will not be translated. In the absence of that regulatory protein, that messenger RNA forms a different secondary structure. Then that ribosome binding site, the Shine-Dalgarno sequence, would be accessible, and the ribosome can bind on there and translate the protein. The last example is something called a ribozyme. So a ribozyme is actually a RNA that has catalytic activity on its own. And what happens in this particular gene is there are two secondary structures that the messenger RNA could take, and one of those causes it to actually cleave itself and break itself down. So this particular gene encodes this RNA, you make this protein, glutamine 6-phosphate amidinotransferase, and the product of that protein is actually a molecule shown in blue here. And this product will bind to the messenger RNA that encodes that enzyme, and when that particular effector is present, that changes the conformation of the RNA and the RNA degrades itself.